Hi everyone. Thank you again for joining us. It has been fun. That's the eighth seminar, and it has been a great success. And we're really delighted to have all of you. It's my pleasure to introduce Corey. And before I introduce Corey Allard, I will just give a kind of a very brief introduction of EpIC, what EpIC is about. It's Electrophysiology and Ion Channels seminar series. It's designed to kind of try to put forward junior investigators, postdocs, and junior assistant professors to share with us their very exciting science. We have had kind of 7 former seminars. You can nominate speakers on the website, which you can check. The link is on the slide down there. We have already a few talks lined up for next year, and we are still building kind of next short while. It's for today, it's my pleasure to introduce Corey Allard. Corey is a postdoc in the lab of the Bologna lab at Harvard. Corey, his major interests have been kind of molecular and cell bases of novel organism physiology and behavior. And as you will hear during his talk, he's interested in specialist and unusual organisms. Corey did his undergrad at Northeastern University in Bachelor of Science in Boston. During this time, I believe he spent 13 months in Antarctica studying cold adapted physics, and probably this might have been kind of part of the inspiration of him interested in studying unusual organisms, as he will share with us today. After that, Corey did his PhD at Dartmouth, and after that, he moved to Harvard to start his postdoc. Since 2019, he has published, and before that as well, he published beautiful work in several publications. He is currently receiving multiple postdoctoral fellowships from NSF and the Charles King Trust Postdoctoral Fellowship, among many other awards. And we are very excited to hear his research. And welcome, Corey, and please take it over.
Thank you, Osama, and thank you to the organizing committee for giving me the chance to talk about some of our work, focusing on interesting examples of sensory innovation across the animal kingdom. And I want to start with really a simple observation that wherever you happen to be zooming in from, we live in a complex world full of possible sensory stimuli. So for example, if you are sighted, you're sensing the light, I guess from your computer screen to form images of my slides. If you're a hearing person, you're sensing vibrations in the air to perceive my words. And you're also probably sensing chemical information, maybe the complex mixture of organic molecules that make up the aroma we recognize as coffee that's helping keep us all awake. And so based on the information in the room that we can sense, we as humans share in perceiving that we are at a Zoom seminar. But these are only the stimuli that we can sense and thus perceive as the world around us. And I think it's important to remember this is not the full world, and it's not an exhaustive list of all the information that's present in the room, if only we had a means to interact with it. And so consider, instead of a seminar for humans, this were a seminar for deer ticks. And so deer ticks don't have eyes, so they would not see my slides. It doesn't have ears, and so it can't hear. And it almost certainly can't smell coffee. But ticks can interact with information in the room in ways that we can't. So for example, instead of eyes, the Haller's organ on the tick forelimb is extremely sensitive to heat sources, such as the infrared that's being released by our bodies. And while they don't have ears, they're still extremely sensitive to vibrations like those we make when we walk or when we talk. And while they can't perceive the chemicals in coffee, they're exquisitely sensitive to the butyric acid coming from your skin and carbon dioxide in your breath. And so these sensory systems are what the tick uses to do the primary behavior that it cares about, which is finding a host. And so while ticks probably don't care very much about Zoom seminars, they are very interested in trying to eat you while you watch. And so instead of ticks, we might imagine a seminar for sea turtles who could sense Earth's gravitational fields and thus perceive cardinal directions, even within the room they're watching from. Or we can imagine a seminar for platypus, which could tell who nearby them is maybe fidgeting by sensing electrical fields generated by their muscle contractions. And so the reason that these diverse organisms would experience this seminar in such profoundly different ways is that evolution has shaped their sensory systems to detect the most relevant information depending on the organism's specific lifestyle or their ecological niche. And so importantly, no organism can or needs to sense everything in their environment. And because of this, an organism's sensory systems often reflect the stimuli that they've evolved to interact with. So what are sensory systems? When I use this term, I'm referring to the molecules, the cells, the tissues, and the neural pathways that an organism uses to detect a given stimulus. And so the flow of information often starts with a stimulus. This could be anything from light to a chemical to heat. And these stimuli are detected by molecular receptors present on the surface of receptor cells. And these cells either directly or indirectly generate electrical responses that communicate sensory information onward to the nervous system to inform behavioral responses. And so as an example, I'll talk quickly about the taste sensory system in humans. And so here our stimuli are chemicals, so sugars for sweet taste, or the amino acid glutamate for umami, or other chemicals like protons or bitterants. And so these, in this case, the molecular receptors are taste receptors. These are proteins that bind the stimulus and generate an electrical response in cells. And so these receptors are found in clusters of specialized receptor cells called taste buds, which are located on a specialized structure called papillae, on a specialized organ called the tongue, that helps facilitate the cells and the molecules do their job, which is to integrate and propagate information they receive on the surface of the tongue onward to centers in the central nervous system that mediate our perception of taste. And so while I'm laying out the basic architecture for the taste sensory system, this is, of course, a gross oversimplification. So while humans can taste sweet, bitter, glutamate or umami, sour, and maybe fat, these are mediated by different molecular receptors like I'm showing you here. But of course, different organisms will have distinct demands for their sense of taste. So for example, cats, which are carnivores, have lost the functionality of their sweet receptors and instead rely on their umami receptors to determine food palatability. Conversely, hummingbirds and songbirds that consume primarily nectar have converted their ancestral umami receptors into additional sweet receptors in the course of their evolution. And this reflects the importance of distinguishing sugars in their diet. And my favorite example, well, taste buds of many organisms are found within their mouths. Catfish have taste buds covering their entire bodies, and they're essentially swimming tongues that are perceiving their external world through their unusual sense of taste. And so I've chosen to talk about taste because it's near and dear to my heart, but other sensory systems are similarly adapted to the diverse needs of different organisms. And so how sensory systems function and adapt to fit these incredibly diverse lifestyles is a major open question in biology, and one that I'm very interested in. And so given the breadth of sensory modalities that exist across nature, exploration of these systems has really benefited from considering both conventional research organisms like fruit flies, mouse, zebrafish, many others that have very well developed experimental toolkits. But we've also benefited from studying unusual organisms that have unique or exaggerated sensory abilities that promise to reveal new biology and conserve principles in sensory physiology. And so I personally think these are important and exciting prospects. Can we use these specialist organisms in comparison to the models? Can we use them to uncover new classes of molecules, new types of cells, and maybe even find novel organs and understand their origins? Can we find new functions for molecules we already know about? And through comparison, how can these organisms challenge our understanding of biology more broadly? And on the other hand, by finding commonalities and conservation, What can we learn about fundamental connections between molecules, cells, organisms, and their behavior? And what can we learn about physiology, evolution, and signal transductionist processes? And so today I'm going to talk about two case studies where we've begun exploring unique and interesting biology to answer these kinds of broad questions. So first, I'll talk about how we're learning from cephalopods, how subtle changes to protein structure can really profoundly impact organismic physiology and behavior. And then I'll talk very briefly at the end about a new direction where we're using unusual fish called sea robins to understand the origin of new traits, especially sensory traits. Okay, so cephalopods first. And so a few years ago, we became interested in the appendages and the sucker cups you'd find on creatures like an octopus here. And these are truly unique organs that are present only among cephalopod mollusks, so octopus, squid, cuttlefish, etc. But they're absent from other mollusks like bivalves and indeed all other animals. And so these structures have long been hypothesized to be sensory organs, and it's based primarily on how the animals use them. So for example, octopus here are voracious benthic predators, and they use those arms to explore the substrate and to probe areas that are inaccessible to their traditional sense organs, for example, their eyes or maybe olfaction. And we think that this behavior with their arms is related to their unusual distributed nervous system. So while octopus are said to have about the same number of neurons as maybe a small mammal, somewhere around 2/3 of those neurons are in the arms themselves. And so the result is that octopus arms have a really high degree of autonomy from the rest of the animal. And so you can see here in this video, there's an octopus blindly reaching through a series of holes in order to capture a crab here that's mounted on the other side of its tank. And so here it seems like octopus arms or maybe the sucker cups are sense organs, but we wanted to know, are they? And if so, how do they work? And so the goal is to fill in the framework that I discussed for a taste sensation, except that we know pretty much nothing at any of these levels. So we don't understand what are the stimuli and what do they tell the octopus about its world. We don't know what the receptor molecules or cells are or how they interface with the rest of the nervous system to mediate behavioral responses to environmental stimuli. So how are we going to study this? How are we going to tackle it? We turned to some early studies where we saw drawings of what looked like sensory cells in the rim of the sucker cup epithelium. So these are spindle-shaped cells, which are often sensory, being synapsed by nerve fibers that run from these large ganglia that sit underneath each sucker cup on the radial nerve of the arm. And so to see if indeed these drawings of cells are sensory cells, we use a strategy where we break up the sucker cup tissue into single cells in a dish, and then we can study them with patch clamp electrophysiology to record electrical signals from cells in response to stimuli. And so here are 3 different cell types that fall out of this prep. And as I hope you can see, 2 of them are spindle-shaped, kind of like those drawings. And we first wanted to ask whether any of these cells might be mechanosensory. And we did this by introducing a small probe to poke the cells while we're doing our recordings. And when we do this, we see that this second cell type here responds to touch with this short, rapidly inactivating inward ion current, suggesting that these cells are mechanosensitivity. And so next we wanted to ask, well, what about chemicals? Are any of these cells chemosensory? And we do this by perfusing the cells with an extract that is derived from prey organisms, so in this case, fish. And we see in this case that we measure an inward flow of ions in this first cell type, indicating that a second separate cell type is responsible for chemosensation. And so here we think we've identified 2 sensory cell types, one that mediates mechanosensitivity and one that mediates chemosensitivity in the rim of the octopus sucker. Okay, so next we wanted to know what are the molecular receptors that endow these cells with their sensory properties. And so to get at this, we use a strategy where we compare gene expression in different octopus tissues. And the logic here is that sensory receptors should be more highly expressed in this rim of the sucker cup compared to the other tissues. And when we did this, the results were somewhat surprising. The only known sensory receptor that fell out of this analysis was the well-established mechanosensor NOMC. So this has been well studied in, for example, flies. And we can use fluorescence RNA probes to visualize NOMC transcripts in spindle-shaped cells in that octopus sucker cup epithelium, suggesting that NOMC is our mechanosensor. But what about chemosensation? So curiously, we couldn't find anything in the transcriptome enriched that resembled a known sensory receptor. There were none of the usual suspects, no taste receptors, no gustatory receptors, no olfactory receptors. But what we did find was this entire family of unusual receptor ion channels that had expression enriched in the sucker rim. And so curiously, these receptors don't resemble known sensory receptors. Instead, they annotate as atypical acetylcholine receptors or neurotransmitter receptors. And so this family, this expansion had been flagged in the 2015 octopus genome. And what was curious about them was that they lack some of the canonical residues that are required for binding and gating by neurotransmitter receptors. And so that led us to wonder, is it possible that these modified neurotransmitter receptors could have been adapted to play a sensory role? And so to see if these receptors actually localize to sensory cells, in this case, we made an antibody that recognized one specific CR here in green. And we used it to perform immunofluorescence microscopy. So I hope you can see that in the rim of the sucker cup cells sitting on this nerve that runs in a circle around the rim are sitting these spindle-shaped sensory cells. Here's a zoom in of what those look like. And so now we're pretty convinced these receptors are in sensory cells, but what do they sense? Do they sense chemicals in prey extracts? And if so, which chemicals are those? And so to test this, we clone the receptors out from octopus cDNA libraries and express them in cell culture, where we don't have very many endogenous receptors. And we can then use patch clamp to study them in relative isolation. So we can screen a large number, patch clamp, so we can then screen a large number of different chemicals and look for those that elicit electrical activity, indicating that those are our agonists of the receptor. And of course, we want to compare those cloned receptors in HEC cells to actual octopus sensory cells, which we can do using patch clamp, again, of dissociated sensory cells from the sucker cup epithelium, and again, measure electrical responses. So when we do this, the results were actually quite surprising at first. Instead of the small molecules usually associated with taste, like amino acids or sugars, we found that CRs responded to these hydrophobic group of chemicals called terpenes. So these are chemicals that are produced by plants, by bacteria, by fungi, many of which you would recognize. These are the molecules that, for example, give pine trees, give grapefruit, and even beer their characteristic aromas. And we saw similar results when we did this with native cells. We saw different cells responded to different combinations of terpenes and other chemicals. So this was initially surprising to us, that the ligands of these chemicals are hydrophobic chemicals. But we actually think this makes a lot of sense now, given how the octopus uses its arms to probe surfaces. So these are molecules that would not be dissolved in water. Instead, they're going to be stuck on surfaces in the marine environment, and the octopus would actually have to physically touch them in order to sense them. And this really resonates with how the octopus actually uses its arms to probe surfaces. And we can show that these chemicals don't just activate cells, but they actually result in neural responses by recording from these axial nerves that run down each of the arms. And so here you can see that when no chemical is added, we just perfuse seawater, nothing happens. If we add fish extract, we get this burst of neural activity. Or if we add the terpene nucotone, this grapefruit smell, we get this huge response. And if we do this for a number of different chemicals, we can see that the selectivity is similar to the isolated receptors. It's these terpenes and related chemicals. And one of the surprising things that we found is in arms that had been amputated from the octopus, they're totally isolated. These CR agonists, these terpenes, elicited autonomous movements in the arms. So here you can see if we just perfuse seawater, not much happens. But if we add fish extract or nuke ketone, this terpene, we get these robust flailing motions from the arms, but not to control chemicals. And again, we see that the selectivity of these behavioral responses matches up nicely with the nerve recordings, matches up nicely with the cloned receptors and the native cells. And finally, we can show that CR agonists are meaningful to the animal because they result in really robust behavioral responses in experiments like this, where here on the right-hand side of the tank, the floor is coated in agarose infused with the CR agonist polygodial, whereas the left-hand side is just infused with seawater. And I'll play the video, and I won't really need to tell you what the result is. The octopus really doesn't like touching that surface. It tries to avoid it pretty dramatically. And we think that, at least in this case, polygodial is probably an aversive or an alarm chemical. And we think that makes sense because this chemical has been isolated from toxic sea slugs. And so it's possible that this particular terpene could actually be a biologically relevant aversive stimulus. And so to summarize our thinking, we've identified separate types of sensory cells in the sucker cup epithelium, those which respond to touch and also diverse chemicals. And these generate distinct responses to stimuli that are propagated first to the distributed nervous system local in the arms, where they seem to mediate autonomous arm behaviors. And then we think propagated onward to the rest of the animal, the rest of the central nervous system, to mediate coordinated organismic behaviors. And so for the chemical sensation, we find that these behaviors are mediated by this new class of receptor ion channels that we call CRs, for chemotactile receptors. And so in doing these studies, we've convinced ourselves that CRs are bona fide sensory receptors, but what are they and where did they come from? And so based just on sequence alignments, we see that CRs are this branch of highly derived nicotinic acetylcholine receptors, as I mentioned. And so curiously, nicotinic receptors are known to play diverse roles in neurotransmission, for example, at the neuromuscular synapse. But we see that CRs are clearly been adapted for a role in environmental sensation. And so to study how CRs have transitioned from this role in neurotransmission to sensation, we wanted to incorporate structural biology to actually look at the receptors. And we did this in collaboration with Ryan Hibbs' group, who's now at UCSD. And so we first solved the cryo-electron microscopy structure of one CR. This is one we named CRT1. And we can immediately see its general features, which are similar to other cis-loop receptors. We see that the complete ion channel is formed, is pentameric. So it's composed of 5 individual subunits that form a complete receptor. And if we zoom in on a particular subunit, we see that it has a clear transmembrane domain and a large extracellular domain. And we can resolve ligand bound at the canonical binding site, which is formed by the interface of 2 subunits. And we can see in this case here the detergent diosgenin sitting in that binding pocket. And so how does this receptor compare to a normal nicotinic receptor? The short answer is they're really incredibly similar. So here on the left, I'm showing an overlay of the extracellular domains of octopus CRT1 in blue and human nicotinic receptor that I'll just call alpha-7 in gray. And I hope you can appreciate that despite these 2 receptors being separated by almost 800 million years of evolution, and these 2 organisms sharing a last common ancestor that looks something like a flatworm, they nearly superimpose. And here I've rotated it 90 degrees so you can stare down the ion channel and appreciate that, yeah, they're quite similar. And so the main question then becomes, how are these 2 receptors that are structurally so incredibly similar, how are they functioning in such profoundly different ways? And I'm going to skip over a lot of experiments we did to show that indeed these receptors are very similar biophysically to each other, to focus in on the differences, which largely comes down to that ligand binding domain formed at the interface of the subunits. And so we see that in alpha-7, in human alpha-7, acetylcholine and other related neurotransmitters get trapped within a cage-like structure. But we see that in the octopus receptor, this cage is totally gone. And instead, we see this, it's indicated in orange, this hydrophobic groove we call a greasy binding pocket, totally replacing this cage. And it's nicely suited to binding hydrophobic molecules like terpenes, for example. And so it seemed to us like adaptation at this ligand binding site is really critical for receptor evolution. And to test this, we aligned all the different sequences of the known CRs that we have identified and compared the variability at each amino acid position to identify residues that are under elevated selective pressure. And what really jumped out at us was that some of the hydrophobic residues in the binding pocket that actually mediate contact with the ligand are experiencing some of the highest rates of diversifying selection across the protein, indicating that indeed this region is really critical for driving these functional differences. So what happens if we mutate one of these important hydrophobic residues? So I'm showing a patch clamp trace on the left. The wild type receptors are activated by these 3 terpenes, these inward ion currents, and they are blocked by the inhibitor mecamylamine. Mutant channels, surprisingly, are actually constitutively activated, and they're largely insensitive to terpenes. They're actually slightly inhibited by them. But the receptors are still blocked by mecamylamine, indicating that these are still functional receptors that are folding properly. They're just being, we just affected gating with these mutants. And so this result really emphasizes to us that these divergent residues are really critical for receptor gating in this case. So in doing these experiments, again, we've started to gain an understanding of what this chemotactile sensory system is and how it's used by identifying and describing these key components at each level of organization. And we were really struck by how the sensory system seems to nicely reflect the physiology and the behavior of the octopus, which is exploring the substrate here. But we wondered whether chemotactile sensation was relevant in other cephalopods, especially ones that have different ecologies, for example, squid and cuttlefish, which can be pelagic, for example, and have 2 additional appendages called catch tentacles that they use in predation. So we looked in both published data sets and did a lot of sequencing of our own. And what we found is that CRs are present only in the lineage of cephalopods we call coleoid cephalopods. So that includes octopus, squid, cuttlefish, but it excludes nautilus, for example, and other basal mollusks indicated by this yellow box. And so to study these other CRs in diverse cephalopods, we turn to the pajama squid, which is sort of our model squid that we can keep in lab. And pajama squid are quite different from octopus. They don't explore the substrate. Instead, they do this behavior here where they'll hide and they will shoot their catch tentacles at passing shrimp in this ambush predation behavior that I'm showing you here. And so we wondered, could CRs, this chemotactile system, play a role in behaviors like this? And so to get at this, we again use RNA sequencing to look for CRs expressed in the squid sucker cups, using a similar approach as the octopus. And we find that indeed squid have CRs, but we find only 3 of them in the pajama squid compared to the over 26, I think we've now identified in octopus. And so we wanted to again identify what the stimuli are for these squid CRs. And so we cloned one of them in this case and studied it in cell culture as with the octopus receptors. And what we found was really striking. Instead of sensing terpenes like the octopus receptors, the squid receptor was responding to bitter compounds like denatonium and to a lesser extent chloroquine. And so we named this receptor CRB for bitter. And so this difference led us to wonder if sensation of bitter chemicals was somehow unique to squid and therefore perhaps uniquely relevant to ambush predation, or if maybe we've just undersampled the receptors if we just haven't looked at enough different ones in both species. And so to determine if bitter sensation is unique to squid, I again use a strategy where I record from the axial nerves of octopus or squid appendages. And that allows me to measure responses to sensory stimuli, regardless of which molecular receptors are being used in the arm. And when I do this, I see that while the arms of both octopus and squid respond to things like prey extracts and indeed terpenes, only the squid appendages responded to bitter compounds like denatonium. And so then we developed a behavioral experiment to test whether or not these chemicals are actually relevant to squid behaviors. And so in this case, we just soak shrimp in these bitter chemicals like denatonium and see what the squid do. And so whereas the squid will rapidly eat a normal shrimp, it doesn't handle it at all beforehand. It just kind of gobbles it. The shrimp soaked in denatonium, the squid will just kind of play with it for a while. It handles it for a really long time before typically ultimately rejecting it. And so we think that in this case, these bitter chemicals are probably relevant for determining food palatability in the squid. And this got us, excited about the idea that CRs maybe function differently in different lineages of cephalopods. And so we wanted to understand this and characterize it a bit more thoroughly. And so what we did was to compare the DNA sequences of all cephalopods CRs in comparison to the related nicotinic receptors and just organize them into this sort of master CR family tree. And what we found was really striking. We found that there are 3 distinct groups of CRs that are present in different cephalopod lineages. So this one here in red, we call CRB for bitter, is the most ancient, and it's found only in decapods like squid and cuttlefish. CRX is more recent, and it's uncharacterized and it's restricted to octopus. We have no idea what this group of receptors does. While there is a shared group, CRT for terpenes, found in both octopus and in decapods like squid and cuttlefish, and it mediates terpene sensation on both. And so we see that the octopus receptors are generally newer in evolutionary time, and there are many more of them. And we think this could reflect the relative complexity of the octopus nervous system compared to squid and cuttlefish. So this also got us wondering, what's different about these diverse receptors? How is it that they're mediating sensation of these different stimuli? And so we again turned to structural biology, again with Ryan Hibb's group. And we did a cryo-EM structure of that squid receptor, CRB, and we can see immediately that it shares a lot of similar features with the octopus receptor. It's a pentamer, and it shares a similar binding site at the subunit interface. But in this case, we can resolve the bitter compound denatonium in this binding site. And when we look closer and looked across receptors, we noticed that there actually appeared to be something of a structural progression, whereby this aromatic cage-like structure that's used for coordinating neurotransmitter binding in the human nicotinic receptor still seems like it's present in the squid receptor. It's just been modified to accommodate these bitter compounds. But this cage-like structure is totally gone in the octopus receptor, and it's been replaced by these hydrophobic residues in this groove that's well suited for hydrophobic molecules like terpenes that you might find on surfaces in the marine environment. And so I'll finish the cephalopod part with what I think is the takeaway message, which is that cephalopods, as diverse and divergent organisms, are a really unique and powerful group of organisms to discover new biology and also get at these conserved principles that I think are really important through comparative approaches. And so, as you might imagine, there are a huge number of open questions that we're still interested in pursuing. One starts with this observation that there are a huge number of CRs, and so gene duplication appears to have played a really critical role in the evolution of this sensory system. But what's really curious to us is that expansion of this receptor family started before cephalopods, before these are sensory receptors. So there are many of these cis loop family receptors that are expanded in earlier mollusks. And so we're wondering, what is it that these receptors are doing? And one idea is that maybe it correlates with the evolution of complex nervous systems in mollusks. And so to study this, I think we really need to work out tissue-specific expression of these different receptors and the functional properties of this receptor family. So a second major interest is the possibility that CRs maybe could play specialized roles even within an organism. So as an example of what I mean, we're wondering if there could be specialized CRs on the male reproductive arm called the hectocotylus. It has a unique morphology and is used for depositing sperm into female octopus. And so could there be specialized CRs that are involved in reproduction? And similarly, we're thinking maybe about those catch tentacles that the decapods are using in predation. Could there be specialized CRs for predation roles? And finally, we're interested in understanding why there are so many more CRs in octopus compared to squid. And one hypothesis is that maybe they're enabling more advanced signal filtration in octopus, and maybe that somehow enables their more complex behaviors. And so I didn't get to talk about it, but we find that CRs don't just form homomers, like I showed you, they don't just form pentamers, but they can instead form heteromers with other CR subunits. And if you just consider that, there's somewhere around 26 CRs in a given octopus, there could be over 100,000 possible heteromers. And we've shown that, at least in some cases, this can change the biophysical properties of the receptor. And we've really only begun to scratch the surface of this complexity and how the octopus makes sense of it. Okay, so we see here from these studies in sort of an unexpected, but I think really profound way, how cephalopods can teach us about how small changes in protein structure can really drive evolution of dramatically different physiology and behavior. And so for the rest of the talk, I'm going to switch gears and talk about a second case study that I hope will be interesting to everyone. It's a little bit less ion channel-y, but where we're finding in a similar way that these unusual fish called sea robins can teach us about the evolution of new organs. And so here's the sea robin, and I want to start by highlighting that the sea robin faces a really big problem, and that is that its favorite food likes to live completely buried in sand, where they're relatively undetectable by organisms that are relying on traditional sensory modalities like vision or olfaction. You can't see these things. And so the sea robin's solution to this problem is very dramatic. It has evolved 6 leg-like appendages that it uses. These are derived from its pectoral fins. And as you can see, it uses them for locomotion, but also for feeling around for prey in the substrate. And if you don't think that movie was cool, you should probably just sign off, because you won't like the rest of my talk. I think it's very cool. And so there's not much known about sea robins, but there's a little early work done, mostly at the Marine Biological Laboratory in Woods Hole, suggesting that these legs are actually sense organs that are sensitive both to touch, but also these prey-related chemicals like amino acids and betaine, for examples. But they could not find a canonical sensory cell type on these legs. There were no olfactory neurons, there are no taste buds, no solitary chemosensory cells, none of the things that you would think would be involved in chemosensation in a fish. And so the hardware is a total mystery. And so we decided to get these fish in lab, and I found that one particular species, this guy here, Prionotus carolinus, is exceptionally good at this pretty remarkable behavior where it can find and dig up prey completely buried in sand. So there's a mussel buried right here under my pointer, and you'll see that this fish is going to dig it up and find it. And they can do this within a few minutes without seeing where anything was buried. They're remarkably good at this. And to be certain this was a naturalistic behavior, we've started filming these guys in the wild. So here's some diver footage where you can see there's a sea robin right here, and it is spending pretty much most of its time really moving sand and digging. And you can even maybe see that there are some other fish following it around, trying to exploit its sensory abilities to steal food that it digs up. So they're really, really remarkable fish. And so we were wondering, how are we going to figure this out? How are we going to understand how the sea robin does this? And we really got pretty lucky because it turns out there are actually 2 local sea robin species that are available. The one I just showed you here in red, Carolinus, is a really robust digger. But in our experiments, we've seen that this other species in blue, Prionotus evolens, it doesn't dig for food at all. Instead, it just walks around on the seafloor. And Oops. So to try and understand this really dramatic difference in behavior, we wanted to look at the leg physiology. And so when we do that, we see right away that this digging species, the tips of its legs are totally covered in these epithelial protrusions that I'm going to refer to as papillae, which we can see are really densely innervated. They're packed full of nervous tissue, and those neurons extend to the surface of the epithelium. But the other species seems to completely lack papillae, and instead it has these broad, shallow ridges in the corresponding locations. And these are innervated, but not nearly as densely. And so our hypothesis here, which is maybe kind of obvious, is that the papillae structures are maybe a key adaptation that could facilitate digging behavior. And I'll just summarize in the last couple minutes here kind of three broad approaches that we've taken to test this hypothesis. And I'll start with the electrophysiology. So we know from the early study that at least some sea robin legs seem to be sense organs that can detect chemicals and mechanical stimuli. So we thought that maybe papillae could result in some kind of sensory specialization that facilitates digging. And so to test this, I developed a prep where we could record from those very large spinal nerves running down each leg. And so I could use calibrated filaments to deliver pokes to these regions of the leg with the different morphology. And I could see that while both legs respond robustly to strong force, like something like a kind of a pinch, only the digging species with these papillae is making responses to very low force, something like an eyelash touching it. And you can imagine that might be useful for distinguishing objects or textures in sand. And to test chemosensory differences between these two species, we again use this whole nerve recording approach. And we see that while both species do have some chemical selectivity, like they both respond to the chemical betaine, the digging species has this really dramatically enhanced chemosensory properties where they're able to sense things like amino acids, common prey-derived tastants, and subsequent behavior experiments have shown that these chemicals that are differentially sensed, the sea robin can use them, the digging sea robin can use them to actually locate, for example, chemical capsules and can find them using those chemicals as cues. And so the hypothesis here is maybe these expanded chemosensory properties that we've discovered could facilitate detection of buried prey. And so based on these experiments, we think that the papillae are mediating enhanced sensitivity to touch and also to chemical cues, and I'll get at how in just a second here, to facilitate digging behavior. So next, I'll talk about how we approach this hypothesis from an evolutionary perspective. And what I mean by that is that we wanted to know if we could leverage the really incredible biodiversity of sea robins to understand their leg physiology. And so what I did was to first consider the phylogenetic tree of sea robins. And so here I'm labeling our two species in red and blue using those same colors. And I hope you can see they're reasonably far apart. And what I wanted to know is, do more of these species look like diggers or do they look like non-diggers? And so to test this, I borrowed museum samples from the Harvard Museum of Comparative Zoology and just looked for papillae on the legs. And so when I do this for most of the species on the list, what I see is pretty striking. The papillae are restricted to this really small lineage of sea robins that are related to the species I showed you digs, where most of the rest of them lack them. And so then we wanted to test, okay, well, does papillae actually equate to digging behavior? Is this the group of diggers? So we were able to use this museum analysis to guide acquisition of live animals. And so we got some additional species that either have papillae, this is another species that has papillae, and some that don't, that lack papillae. And when we put them through our behavioral and our electrical physiological pipeline, we see that the papillae is really correlating with the behavior, that if you have papillae, you dig and you have these enhanced sensory properties. And if you don't have papillae, you don't dig and you don't have these enhanced sensory properties. And so it really matches up nicely. So here we think the observation that papillae are only found on the legs of these digging sea robins is again strong evidence that this is an adaptation for digging. And then finally, I'll talk about some evidence that comes from our analysis of sea robin development. And so one of the things I think is really incredible about sea robins is that they are born without these leg structures. Instead, so when the sea robin is born, it has this fused pectoral fin, looks like a normal fish, and the legs then separate, the first three rays separate during the first month of development to give rise to the legs. And so when we looked a little bit closer, we could see that there was actually a period of time when fish have legs but don't have papillae, and that they then gain the papillae later. And so we could then use this to test the importance of those papillae, both in the behavioral assays and using electrophysiology. And what we could see is that indeed the occurrence of digging behavior correlates not with the emergence of legs. So when they have legs that look like this, they're not digging. It's correlating with the emergence of papillae on those legs later that they start digging. And so again, this really drives home the idea that papillae are important to us, are important to digging. And I'll finish with just a quick overview of what we've learned about the molecular and cellular basis for these unusual chemosensory system. So we took advantage of the observation that it's really the distal legs where we find those papillae that are chemosensory, but the upper part of the leg doesn't seem to be. And so we could use this to do RNA sequencing, comparing receptor expression in the distal part of the leg versus the proximal. And what really jumped out at us was that the most enriched gene in the distal part of the leg is a taste receptor, is this taste receptor T1R3. And so we wanted to know if it localizes to papillae. And so we again use an RNA probe, a fluorescent RNA probe, to visualize the receptors. And we can see indeed that the receptor seems to localize to the tips of those papillae, the pink staining here. And so it seems like there are taste receptors on the tips of those papillae. And we see that they're absent from corresponding regions in the non-digging sea robin. And so to understand if these taste receptors could actually mediate sensitivity of the types of chemicals that are uniquely sensed by the digging species, we teamed up with Maud Baldwin's group at the Max Planck Institute to test the selectivity of these receptors in cell culture. And indeed, we see that they are a really good match for the chemicals like the L-amino acids here that are sensed by the legs of the digging species specifically. So this gives us a clue, but what are these cells on the tips of the papillae and how do they work? We really still don't know. So you'll have to tune into a future talk sometime to find out. And I'll end here with the future directions for the sea robin work, first and foremost, which is of course to better understand what are the sensory cells and molecules that are endowing the legs with sensory properties. And we can then investigate, what about these molecules and cells is specialized in the digging sea robin to enhance sensitivity. In parallel, we're looking at developmental questions. So how do the legs form in the first place? How are papillae patterned during development? How do they form? What is the genetic basis? And then finally, we know that the evolution of these leg-like structures was accompanied by really dramatic changes to the sea robin neural architecture. So they have these really massive leg-specific spinal ganglia that are many times larger than any other spinal ganglia in the animal. And they also have these additional lobes of the spinal cord that are dedicated to, presumably, to processing sensory information coming from the legs. But we have no idea how this hardware functions, where it comes from, how it's wired to allow the fish to respond to this critical new sensory information coming from these novel organs. And so that I think is a major future direction for this work. And I'll end with what I hope is the takeaway message, if you remember nothing else. I think that comparative strategies featuring sensory specialists, like sea robins and like cephalopods, are a really powerful approach for uncovering new biology and conserved principles. And I think that sensory systems especially are really excellent and clear models for studying connections involving basic biology, basic connections between protein cells, organisms and their behaviors. And with that, I would like to thank you all for coming and for your attention, and the EpIC committee for hosting me. And also my lab and collaborators and Ryan Hibbs' group, who really contributed to the cephalopod work, and I've highlighted these people in orange. And to my lab members and collaborators, including Amy Herbert, who's a postdoc with David Kingsley and really has pioneered a lot of early genetic work in C. robins, and Agnese Seminara in Italy, who have all been instrumental in this project. So with that, I'd like to take questions if anyone has them.
Thanks, Corey. Great talk. Really enjoyed it. And we have a few questions here, and I anticipate we might get some more. So I'll start with the first question from Paul Fluffman. Any particular reason why terpenes would be specific for fish living or decaying?
Yeah, it's really interesting to us. You know, a lot of terpenes are in a terrestrial environment. They're volatiles. They're types of things that we would smell with our olfactory system and would be relevant in say distance chemosensation. You know, in the aquatic environment, of course, they're hydrophobic. They're going to be on surfaces. And so far, we think that most of the chemicals we've identified or tested, the animals don't seem to like them. They're possibly alarm cues. And so our maybe oversimplified way of thinking about it is as they're reaching into these cracks, for example, crevices in the seafloor, if there's something dangerous like a toxic sea slug, that's going to indicate to them, like, don't get your arms out of there. We've also been identifying new types of chemicals that are activated by this system that maybe could play different roles. So we found that hormones are agonists of certain receptors. We found bile acids, pH even for certain receptors. So we're really just starting to get a handle on the logic of the whole system of what the chemicals mean to the animal and how they're encoded and which receptors are involved. So it's a good question, and I don't have a comprehensive answer for you, I'm afraid.
Yeah, related to the same question, I think. What's the favorite taste for squid and octopus?
So it's actually kind of a funny story. The maybe it's not the favorite taste, but it's the most potent agonist of these receptors that we identified was by accident when we were trying to do the cryo-electron microscopy. We were having trouble getting a structure that had the ligand bound. And what we noticed was that there seemed to be a detergent stuck in what we thought was the binding site. And so we did a number of experiments. We tested that detergent and whether or not it can activate the receptor. And it is the most potent agonist that we've identified to date, is the detergent we were trying to use to purify the proteins for cryo-EM. So kind of maybe also getting a little bit at the purpose of these. That's another, you know, at least partially hydrophobic molecule, a surfactant.
Sure. Yeah, that sounds good. Another question from Rohit Kumar. Were you able to create any specific aversion to the compounds by neuronal stimulation?
So I don't think I fully grasped that. What was the question again?
Were you able to create any aversion to the compounds by neuronal stimulation?
By neuronal stimulation. If I'm understanding the question right, so most of the compounds that we've tested seem to be aversive. We don't have, actually, we're still trying to find something that seems appetitive. So I'm not sure if that answers the question or not, if maybe I missed some nuance, but pretty much everything seems to be aversive innately so far from what we've tested.
Mark Rogers asked, vertebrate nicotinic receptors frequently form alpha-beta subunit terminals apart from alpha-7 monomer. So do you have any data on potential for alpha and beta CRs in cephalopods?
Yeah, so that's a really great question. Can we borrow from the what we know about nicotinic receptors to understand how these function? I think that there almost will be something analogous to that arrangement. We do see that there are, for example, receptors that seem silent and maybe could play auxiliary roles, and so we're testing that sort of thing now, trying to understand what heteromers actually form and what their function is and what the role of the individual subunits are. But I think that's a really good point and one that we're considering.
That's good. Considering the sea environment, do you know, which is very different from non-sea environment, to what extent do the electrical properties kind of change, like channel conductance, receptor sensitivity, etc.?
Yeah, we've thought about that, and I don't have a good answer, but I think it's really interesting, how um, how is gating differently? How does the receptor work differently in seawater um, than in a lower osmolarity solution? And it's something we're thinking about, but I really don't actually have a good answer for it. Right now, we don't know.
Theon Frampton, thanks very much for sharing such fascinating work. Forgive me if the question is a bit ignorant. I'm no expert on octopus physiology. My understanding is that their legs are relatively autonomous. I'm wondering, do you think the octopus that touches a surface coated with a nasty paste is perceiving the sensation in its CNS, or is the reaction of the arms more like a reflex, essentially like gagging when you taste something bad?
Yeah, so uh, we haven't tested this exhaustively, but my guess based on what we have seen is that it's a combination, that a lot of those behaviors are sort of hard coded locally in the arm, and that there's maybe we're hoping to find some like stereotype reactions to certain chemicals that maybe is just helping, this arm over here respond kind of on its own, where this arm over here might be totally fine. But we do see that the animal behaviors will be different. The arms don't necessarily just spasm when the intact animal encounters one of these cue. It can maybe even suppress that response. Is kind of how I think about it. That it's coordinating these autonomous arm motions, but that's all things that we're thinking about going forward. Yeah.
Sure. Bertil tells you, great talk, thanks. Chris Ahern, in human acetylcholine receptors, aromatic tryptyrosine alanine residues in the binding pocket appear to utilize electrostatic acids, cation pi, to bind ligands. Do you think that these evolved pockets could also bind more divergent chemistries outside of these hydrophobic terpenes?
Yeah, we definitely think they do. And we're starting to get a handle on that, both by screening different types of chemicals, but also trying to identify by natural compounds that we are trying to purify out from, for example, prey mixtures, or from actually we're looking at a lot of bacteria now that we culture from different surfaces in the octopus's environment, and we do find that they're very promiscuous in terms of their binding, and that pH plays a major role in that effect. Yeah, another thing that a lot of my answers will be, I don't really know what we're thinking about it.
Yeah, it's
And this is another one.
scratching the surface. So
Yeah.
that's great. There are many other things, but the last question that I can see is CRT1 seems it doesn't have a structured C lobe, or it seems to be shorter than alpha 7R. And the receptor response to different compounds is clearly different between these two receptors. How is the mechanism of activation after hydrophobic ligand binding?
What is the mechanism of activation? So we're not entirely sure that the person who posed that is right. They're missing that part of the C loop. And so the gating is one thing that we think is strikingly different. So especially in the octopus receptor, if you make similar mutations to the ones we did in like human alpha 7, or even in the squid receptors, you don't get constitutive activation. You get a loss of binding, and so there's differences that we don't fully understand. The other thing that I haven't talked about, because we don't understand it at all, is a number of octopus receptors, including CRT1, is voltage sensitive. It's gated by hyperpolarized potentials, and that seems to change ligand selectivity to some degree. There's an interaction between chemical binding and voltage sensitivity that we don't understand, but we think is really interesting, because maybe that could be a mechanism for encoding different chemicals, or something like that, or changing spike patterns if it's chemical 1 versus chemical 2. Again, don't understand it, but we think it's very interesting.
Yeah. Last question from Damien Bell. Why is it all aversive? Do you know why everything not attractive?
Yeah, so again, this is one that we think about, but where I can only kind of wave my arms. So what I will say is, if you do something like put a rubber crab in an octopus tank, it will try to eat that rubber crab. And so it almost, maybe an oversimplified explanation, I'm sure this is wrong or too simple, is that maybe the octopus's default is just to eat unless there is some signal that says don't eat. And that kind of pans out in some of our really oversimplified experiments. And I'm sure all the other octopus people out there will hate me for saying it's so simple. That's one idea. Everyone's free to try and prove me wrong that octopus are eating everything unless there is a no queue. And we're looking really hard to try and
unless
find.
proven otherwise.
Yeah.
Yeah.
So
looks like the very last question, because we are already 3 minutes beyond. Maybe I missed this. Do non-digging species lack taste receptors in their genomes? If you were to topically express these receptors in the arms, would they learn to dig?
So they have them in the genomes. We can even visualize them in the mouth, for example. They have the same complement of taste receptors as the digging species. They're just not expressed in the leg tissue that we can see.
Cool. That sounds great. This is awesome. We're already a few minutes after the hour, but thank you so much. It was really fun and exciting for many people, and as demonstrated by the many questions we have been getting. So thank you again, and all the best with your research. Looking forward to hearing more about your future research.
Thanks very much. Thanks everyone for coming.
Thanks, Corey.